This episode is supported by two companies directly invested in advancing GPCR research, Eurofins DiscoverX and GeneTex. Eurofins DiscoverX brings decades of validated GPCR assay expertise to teams building real drug discovery programs. It's trusted across discovery, development, and regulatory review. GeneTex believes antibody trust is earned, not claimed, proving it through rigorous validation and an open invitation to the community to test it, the community being you. You get to challenge and give feedback. Request your free anti-GPCR antibody sample today. Links to both are in the show notes. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR. I'm very excited to be recording this particular podcast episode. Joe reached out to me on LinkedIn, and we started a fantastic conversation. And I'm excited to bring you, Joe, to the podcast today. Joe Kim, welcome to the Dr. GPCR podcast.
Thank you, Yamina. It's an absolute pleasure to be here.
Very excited to have you on. Maybe we can start by you introducing yourself to the audience. So tell us who you are and what is it that you do.
Of course. So my name is Joseph Kim. I go by Joe. I am currently a postdoctoral scholar in Professor Ashish Manglik's lab here at the University of California, San Francisco.
Wonderful, wonderful. And I'm going to put it out there on the record. I've been reaching out to Ashish. We're going to have to push him towards coming onto the podcast. I don't know if it's ever going to happen, but I actually listened to one of his talks at a conference, at a Gordon conference, and he was just phenomenal. So Ashish, if you're listening to this, we really want to hear from you on the podcast. So Joe, tell me a little bit more about how did you end up working on GPCRs in Ashish's lab? I'm curious about your career trajectory up until today.
Of course. I guess, should I start from the beginning or just the moment that led me to get into GPCR research?
Wherever you want to start. I think, for example, we've had guests who went into details and said, as a child, this is what I liked. We had a lot of people who liked astronomy and ended up being GPCR pharmacologists. There are people who lived on a farm and ended up working in banking to then end up working on GPCR. So you can start wherever you feel comfortable.
I see. Okay. So I guess then, this will go directly in line with some of the topics that I know you and your guests like to talk about. But when it comes to how I got involved in GPCRs, so since I was a kid, I went to the library a lot. And I also grew up in Corvallis, Oregon, which is like a college town 90 miles south of Portland. And there were a lot of trees and outdoor flora and fauna that were surrounding me. So I got a chance to kind of play around the trees and explore all the various fruits and vegetables that were growing around. And when that kind of connected, it kind of drew me to sort of say a book that I think I encountered one day in the library that talks about, this is a berry bush that you can eat from, and this is a berry bush that you really shouldn't eat from. And so I guess my interest in drug research started with poisons, and then there's that whole saying that the line between drugs and poison are not really so different. And so I've always had this fascination with drug research since. And I mean, GPCRs are indeed a really prominent drug target, which I know is a question that you like to ask to your guests quite often. So prematurely answer, yes, I definitely do believe that GPCRs are good drug targets. So, and then what happened actually is I didn't think back to that moment until I was in my final year of graduate school at UW-Madison. I was thinking about postdoctoral positions. I knew I still wanted to continue in research, but I wanted to directly get involved in drug discovery projects and groups. And I was just reading through a whole bunch of different papers. And then one day, I remember this moment very distinctly. And it was, I think, in September of 2022 or 2023, where I was just reading through a bunch of papers, and then I kind of sat them down and I kind of exclaimed to myself, crud, I'm going to have to study GPCRs, aren't I? And that's how I ended up here, because I wanted to stay on the West Coast, but I also wanted to look at which groups were really involved with drug research and also involving membrane proteins. And lo and behold, Ashish's lab was definitely high up there on my list.
Wonderful. I'm curious about how did you end up in science in general? Like did you have, I don't know, were you inspired by a teacher, a math maybe, or a chemistry teacher, or how did you choose the scientific path initially?
So I think the scientific path that I chose, I think I've always had an interest in science to some degree, particularly based on sort of that poisons book that I was telling you about. That kind of always got me interested in sort of the health sciences itself. And I kind of held on to sort of that on the back of my subconscious as I went through, say, elementary school, middle school, high school, and in college, which I attended at Oregon State University. I went in as a pre-medical major, but then I realized that pre-med itself, or just a career as a physician, wasn't really in the cards for me, because I think a big realization that I came across is that I will spend too much time trying to dig into sort of the why of some treatments are prescribed and or drugs or procedures are done. And I think, I mean, that's kind of the hallmark of a scientist, is it not? Just constantly asking questions of things that we do and or and curious on. So I think it's just always been there, is kind of the short answer.
I love it. I love it. And how did you pick your PhD lab? Was there anything in particular, any topics in particular that you were interested in?
So say, I know I was a very visual person. So, and I can kind of parse out details if I look at a photograph, or in my PhD career, a micrograph. And so at the time as well, UW-Madison was starting up its cryo-electron microscopy facility. And that was how I found my advisor, Professor Elizabeth Wright, because she was hired from Emory to serve as the director of that cryo-EM facility. And so I was her first graduate student to join her lab after she moved her lab from Emory at UW-Madison.
Mm-hmm. Wonderful. Wonderful. And once you were finishing or getting closer to finishing your PhD, what were some of the steps that you took to look for your postdoc? I know you mentioned that you really wanted to stay on the West Coast, and that was one criteria. What other criteria were there when you were, in a sense, shopping or putting yourself out there to find?
Yeah. So I think there was both a level of comfort that I wanted to keep emphasizing, which was cryo-EM. That was my training, but it was more of a subset of cryo-EM known as cryo-electron tomography, where instead of, say, single snapshots of proteins embedded in ice, it was more like a whole series of tilted images of one particular sample. But I wanted to go back, well, not necessarily go back. I wanted to solve structures of clinically relevant druggable proteins. And I was pretty familiar with the technology as well as the methodology of cryo-EM itself. So that was one criteria. The other criteria is, throughout graduate school, I've always had a fascination with membrane proteins, just as an outsider looking in. I think part of that reason is it's kind of, people that know me will kind of chuckle a little bit when I describe this, but I have a tendency, or problem, I guess, of if someone says that this field of study or just any topic in general is quite difficult and or challenging, I will not run away from it. I will run towards it. And membrane protein structural biology was and is still considered to be quite challenging proteins to solve structures of. So those two combinations, combined with that drug discovery aspect that I was interested in throughout my life, kind of led me to really narrow down the list of the labs that I was thinking about.
I think it's interesting that you mentioned, the fact when you think about difficult disciplines or hard to master techniques, for example, is something that you run towards. It's interesting because in the beginning, when we started chatting, you had mentioned that you've always wondered why. And that's what drove you into research and away from medical school. And I think those two components, the running towards or working and difficult to master or a difficult field, and your need to find an answer why are the combination of basically who you are as a scientist.
Before we continue, I want to ask you something. A lot of you listen regularly to the podcast but haven't subscribed yet. If you enjoy these conversations, hit subscribe now and leave us a review. It helps other GPCR scientists find the podcast. And speaking of community, a couple of things worth mentioning before we continue. The Dr. GPCR University is where GPCR training meets a real scientific mission, expert-led, live and on demand, and built to move the field forward. And if you want to go deeper into receptor pharmacology and drug discovery, Terry's Corner is where serious pharmacologists sharpen their thinking alongside Dr. Terry Kanak. It's not a content library, it's a room where the standard is significantly higher. Thank you to GeneTex and Eurofins DiscoverX for being part of this community. Sign up to the Kanak in Brief, the Terry's Corner newsletter, to stay in the know about live Ask Me Anything sessions, live courses, and on-demand lessons coming out regularly. And join the Dr. GPCR ecosystem for upcoming live and on-demand masterclasses and community updates and so much more. All the links are in the show notes. And now let's get back to this episode.
People would say that would make life challenging per se, but I would argue back, doesn't make it boring. That is for certain.
I agree. I think we're both on the same page. I feel like when somebody says, "Oh, this is difficult" or "This is impossible," then I too think about it as a challenge and say, "Well, what's so impossible about it?" And then I typically also think about digging deeper and figuring out a way to systematically address that question and find ways that are logical in order to break that big question down into a smaller subset of questions that you can start or I can start answering one by one. And it's the combination of those answers that give, that big elephant.
And sometimes each of those questions themselves, the way you answer them, may sprout out into other different kinds of questions that suddenly have you exploring topics that you never thought that you would explore.
Yeah.
And then it kind of spirals out in the best way possible.
I agree. I absolutely agree. That being said, if you have a high level question, you should always keep that in mind, because otherwise you might, go into different directions where you can find more questions, get those answers, and then diverge. But I feel like, especially when you're a postdoc, you have a limited amount of time. You have to focus.
Yes.
Focus becomes very important as well. I guess you mentioned what were the criteria for you to look for your postdoc lab in practice. And I'm asking this question for us to understand how did you go about it in a practical manner, but also I'm thinking about anyone listening who's finishing up a PhD and thinking about, okay, I want to do a postdoc. How do I go about it in a practical manner? What are some of the steps that you have taken in order to look for a postdoc position?
Yeah, that's a great question, Yamina. So some of the steps that I remember taking was, no one will ever argue against list compiling of the various labs that you're thinking about. Maybe include a pros or a con section as well. And then I think I just ended up cold emailing a lot of PIs and just asking them, I'm interested. Here is what I have done previously. Here is what I'm interested in doing in the future. And even before sending out that email, I took the time to kind of formulate those thoughts, as well as what I would want to do after my postdoctoral experience is finished. That just compiles into a really nice letter of interest that you can then send over along with your CV. And sometimes they respond back. Sometimes you yourself need to follow up with them about, say, give or take a week after you send that initial email, because PIs are getting emails constantly. And so they might have just missed. So there's definitely an element of proactivity that will reward you in the long run if this is sort of the path that you are thinking about.
I think that's very valuable. And if I can add to that, two things. Number one, I did not do it like that at the time. I talked to my PhD supervisor who said, "Oh, you should look into this lab," or "Oh, you should look into that lab." Now, with my head today, the way I would suggest going about it is, to your point, figure out what are the criteria that you must have, what are the things that you do not want. Some people prefer smaller labs, other people feel better or prefer larger labs. That's number one. Number two, in this, let's say protocol, is approach it as a project.
Yes.
And the checklist is always a nice way to go about it. And I really like, Joe, that you mentioned being proactive. On the receiving end of these emails, there are scientists, PIs, who are very busy, who receive countless emails. And if you find a lab that you're really interested in, I don't think anyone will hold it against you if you picked up the phone and said, I emailed you, I really, 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 really want to work with you. Would you have 10 minutes or 15 minutes of your time to help answer some of these questions?
Yeah, I would 100% agree with you on that.
I also think that if the response to your enthusiasm, obviously stay courteous, but the response to the enthusiasm is negative, that's a sign that maybe that lab is not for you.
And springing off of that as well, I would say, and you kind of mentioned this earlier as well, of kind of diversify the kinds of labs that you would be interested in joining, not only from the scientific research standpoint, but also the size of the lab, the funding situation of the lab, how long the PI themselves have been working within the field, and what is sort of the network that they represent. Just because, say, if you do move on to the next step, which is the initial conversation, whether by phone or Zoom. And then if you move on a step further than that, which is they will fly you out to do the in-person interview / tour, and you find that the environment is not as suitable for your interests as you anticipate, well, then you have fallbacks of different kinds of environments that you get to take the time to explore, and that will help you kind of tailor what your interest and environment would be ideal for you.
I agree. I absolutely agree with what you just said. And if I can add to that, I think it's about about introspection and taking that introspection into an organized, again, this is me trying to organize the world around me into an organized manner so that systematically, you know, what is your next step. And to your point, Joe, you reach out, you go to visit the lab, you like them, but not enough. Then you have the opportunity to say, well, no, this is not for me. And it's okay to say no.
Yes.
And then move on and find the next lab. This is actually wonderful. And if you can maybe share with us, when you interviewed in your current lab, what were some of the things that caught your eye, that you really liked about the team, Ashish, the area? Obviously, you wanted to stay on the West Coast. So I have a sense that you knew what you were getting yourself into from a regional perspective. But I really want to hear, what were the things that caught your eye that you can remember from those interactions early on that said to you, oh, this is it, I really want to be here?
Yeah, thank you for asking that. So I would say things that really stood out to me while I was interviewing in Ashish's lab, as well as actually UCSF itself. So fun fact, when I was interviewing for graduate school, UCSF was one of the places that I interviewed at, but I ended up not getting accepted into their program. So it was kind of like a very interesting return per se. But as to Ashish's lab itself, I think what I admired the most is the environment that I was observing, of where everyone that was working within the lab seemed to be striking that really nice balance of very friendly. They're courteous with each other. They're more than willing to share details about what they're doing / what kind of expertise they're bringing, their backgrounds. But then there's also a great level of professionalism within the lab that I greatly admire. And that kind of makes it really efficient in my mind of sort of the research progress that I want to do, and the strides that I would make when I myself am in that environment. So that's one aspect. The other aspect is there was just so many different projects that were going on in Ashish's lab that I found myself almost, I don't want to say getting lost, more like there's just so much around me that my interest will always be piqued in some way. There are people that are working on antibodies. There are people that are working with GPCRs that is natively purified. There are people that are working with GPCRs that are expressed by transfection, whether that's involved with opioids, whether it's some other different protein family like transporters that are involved in iron. And there's also projects that are involved in cancer research. There's just so much, and I will never get bored when I hear about these different kinds of projects, because when things are different, you have to engage your brain in different ways, and that means different kinds of levels of stimulation that makes it so that you're always thinking differently, and that sort of orthogonal thought process will do no harm to you. In fact, it will help elevate and accelerate the kinds of thinking that you need to apply to succeed in your scientific research.
I think it's wonderful. It sounds like you found the place where, as you mentioned, you would never get bored, and it gave you the sense of excitement of being able to talk to people who work on these different projects, learn from them, and potentially, bring some of those elements into your own project that you are working on.
Yes, and this is something that Ashish himself actively encourages amongst all of us. If, say, like just the other day, I gave lab meeting just this week, and then afterwards, one of my colleagues just came up to me to ask me a couple of questions regarding my project, and now we're gonna team up to try to see if his project is compatible with mine in a simple experiment next week. And if it works, great. We have something new going on. If it doesn't work, well, we learned something. And maybe my colleague could use that to sort of refine his planning on the next way in which he would want to move forward with the research question.
That is wonderful. You mentioned in the beginning, I'm shifting a little bit gears towards the science of it all. You mentioned your fascination with membrane proteins, and you gave us a little bit of an overview as to why you're fascinated with membrane proteins. Can you maybe expand on that and tell us a little bit more about if there's a moment in time where you said, "Oh God, I love membrane proteins because XYZ," or if there is any turning point in your career where membrane proteins entered and started preoccupying the why that you are programmed to keep on asking.
Yeah. So I think I'm not sure if there was an exact moment that I remember where membrane proteins really entered my sphere of interest, but I know one thing for sure, which is that by honestly the middle of year one of graduate school, membrane proteins was always something that was kind of at the back of my mind that I was always kind of looking out for. Year one of graduate school, I think, was around 2018 or 2019. That was sort of when cryo-EM was just beyond its peak excitement per se, because the Nobel Prize was awarded just a year prior.
Yeah.
And additionally, one of the guest lecturers during my undergraduate year who inspired me to enter the cryo-EM field itself was applying it for membrane proteins. I think he was working on connexin hemichannels, which are, I believe, the channel proteins that connect through epithelial cells, as an example, that allows exchange of certain nutrients or ions across and between those two cells, as an example. So how that, and then just thinking about it more, I think membrane proteins for me sort of represent the real, not real, the best way of sort of representing the key and the lock mentality.
Mm-hmm.
In the sense that there is sort of there's a peptide, there's a small molecule floating around in your bloodstream, as an example, and it wants to exert its physiological effects. And most of the time, some small molecules can pass directly through the cell membrane, but a lot of times the small molecule or peptide has to sort of bind to the membrane protein in order to say, hey, this is the effect that I am designed for. Membrane protein, please relay this message that I am carrying. And that's how the downstream processes start, and that's how messages are relayed. And it's kind of the start of it all, and it's just really fun to think about.
I agree. It reminds me of Terry Kanakin, who always says that GPCRs are the Swiss Army knife of physiology. And I think we can even expand that, is that each family of membrane proteins is the Swiss Army knife of physiology. And it's a combination of those that make it so that all of these processes, all of these signals get into the cells, get translated so that the cells can then later on respond.
Yes, and I will comment that sometimes these downstream processes could be an entire spider web network of its own that would really confuse a lot of people, myself included, I think. And this is why initially GPCRs definitely scared me more, because in undergraduate, there was that example of me thinking about and reading about GPCRs, and then somehow it connected to, like, say, the MAP kinase pathway, where it's the MAP kinase, and then it's the MAP kinase kinase, and then it's the MAP kinase kinase kinase. And that would be any sophomore student's nightmare if they kind of have to systematically pick apart those kinds of details during an exam.
No, I agree. But let's also establish that as scientists, we're not very good at giving names to proteins.
Not unless you're in the Drosophila field, in which case that gets fun.
Yes, I agree with that. Absolutely. So you told us a little bit about how you ended up in science, where the fascination to membrane proteins comes when you started working on GPCRs. In your current project that you're working on, I wonder what can you tell us what got you interested in the current project, and is it something that was already established in the lab, or is it something that you kind of built together with Ashish of the team?
So it's definitely both. The current project that I am building / Ashish and I formulated, I can't talk about yet just because it's in its heyday, but I can talk about sort of one of the very first projects that I ever did when I entered Ashish's lab. So background context. I have never purified a membrane protein before entering Ashish's lab. So the training project that Ashish gave me was, okay, we have some small molecule candidates that are exerting really interesting in vivo effects in mice that were found by docking in collaboration with Dr. Brian Scheukett's lab, which I know you've had as a guest before. Yeah. So it was my way of getting trained in membrane protein biochemistry, and this was through the targets of the mu opioid receptor and the kappa opioid receptor. They found a small molecule that kind of acts as an antagonist for mu and an inverse agonist for kappa. And yeah.
Isn't it beautiful? I mean, seriously, it's just phenomenal.
Just when you think pharmacology itself is fun enough, then you suddenly have polypharmacology, and you have this whole other different expanse of networks. But so what happened was I purified the mu opioid receptor and the kappa opioid receptor, bound to that candidate small molecule. And the idea was learn the biochemistry and solve the structure of both GPCRs with those candidates. And it just worked. Like the first time that I was being taught the ropes by Christian, who's been in Ashish's lab for a very long time, it just happened that, oh, I managed to purify this protein in good enough quantity. Okay, let's put it on cryo-EM grids. Oh, hey, look, the particles look pretty well distributed. Let's collect a data set. Oh, hey, this data set looks really nice. Can we get the structure? Oh my gosh, we can see the small molecule. And it's kind of aligned in this way, and it's interacting with this residue. And can lightning strike twice? Can we do this with mu? Yeah, and it ended up happening with mu as well. And things just fell in line, and it was great.
Do you think it's beginner's luck? I have to ask.
I think so, for sure. Combined with the really good training that I received. And I myself told Ashish throughout my interview that I was very afraid of entering the membrane protein biochemistry / structural biology realm. So I needed really in-depth training. And maybe partly motivated by fear, maybe partly motivated by excitement, maybe partly motivated by me just wanting to really push forward as much as I can in this project. I think it was just a combination of all of those factors that really helped me in the end.
I think it's a wonderful story, and thank you for sharing that with us. I think, there's hard work, there's luck, there's the training that you received, and there is also the fact that the molecules that you actually used were well optimized enough that allowed to stabilize those complexes with the mu and the kappa opioid receptors. And I think it's like when the stars align and things just go smoothly.
Yes, without a doubt. And prior to me entering the picture, Ashish's lab kind of developed a really nice pipeline that allows them to purify GPCRs locked in the inactive state for cryo-EM structure solving by a whole bunch of different fabs, nanobodies, nanobody that's anti-fab, etc., that was published by a lab in Stanford right prior. And I entered the picture just as that process was perfected. So wasn't that? Yeah, the toolbox, exactly.
That's wonderful. What happened to that piece of work, to that project? Was it ever published, or?
Oh, yeah, it got published this year in ACS Central Science. Yes.
Congratulations.
Thank you very much.
That is wonderful. All right. I always ask this from everyone. What is your favorite GPCR? That's why I wanted to hear a little bit more about the projects that you're working on so that we can, get to this question. Do you have at all a favorite GPCR?
So people are going to think that it's going to be one of the opioid receptors. I am going to surprise them by saying it's not the opioid receptors, just because I have another GPCR in mind that is much more unknown, that I am always just thinking about wanting to form a project on. And as a matter of fact, one of my projects does involve this receptor. It's known as the galanin receptor. Have you heard of that one?
I'm not familiar.
Exactly. So the galanin receptor, there's a family of 3 receptors, galanin receptor 1, 2, and 3. The first one, galanin receptor 1, is the one that I'm most interested in. It is highly expressed in the brain and the spinal cord. There has been some evidence to suggest that it's involved with, say, feeding, analgesia, similar to the opioid receptors. And as a matter of fact, some papers have reported that the galanin receptors have interactions with the mu opioid receptor. And some labs further than that believe that there's sort of a dynamic interaction that's going on between the two. That being said, and I think this is kind of the crux of the whole issue, the reason why the galanin receptor is not so well studied currently is that no one has been able to find a really potent small molecule for those receptors. And there was a recent couple of structure publications of galanin receptors that's bound to its endogenous agonist, galanin, the peptide itself, and that kind of suggests or hints as to why this may be. The peptide itself is buried laterally in the receptor.
Wow.
Yeah, and we think that may be why we're having such difficulty finding a small molecule for it, because it opens up a whole different new realm of like GPCRs involved in neurology, which is always going to be a topic of great interest to both many groups and many companies.
Wonderful. Thank you for sharing that. I actually, while you were mentioning this, I quickly Googled it, and it looks like, well, it's a biologically active neuropeptide, as you mentioned, but it does function through 3 different GPCRs, 3 different receptors. Do we know anything about the differences between these receptors? Obviously, I can read the paper, but I have you.
I think the main difference seems to be the pattern of expression. I think maybe.
Interesting. I mean, I will read it. It's a PNAS paper. It's called Structural Insights into Galanin Receptor Signaling, published in 2022.
Yes.
And every time, and thank you for sharing this. I'm going to read into this because I always like to learn new things. Every time I hear peptide and GPCR and binding, I think hydrophobic, difficult to access, binding site. Good luck, small molecules.
Yeah. And now you have another layer on top of that because it's the most unusual binding mode of the endogenous agonist. So you can imagine how something exogenous is going to have a not so easy time getting in.
Exactly. Unless you want to think about it, think about drugging it with small molecules with allosterically and modulating the natural peptide's function that way.
And then when it comes to allosteric modulators, you now have a whole different other can of worms that you need to think about.
Absolutely, absolutely. Especially that it's one peptide, 3 different receptors. The receptors seem to be coupling to different G proteins.
Mm-hmm.
And I'm assuming since it's expressed in the brain, it's going to be very difficult to really do an in-depth analysis of the signaling pathways in a native system. So screening becomes, I mean, we're complicating things, but I think this is the type of problems and the type of questions that you find interesting, and I also find very interesting.
And the fun part is you can kind of do that with just, you pick any GPCR, even the most well-studied ones, the beta-2 adrenergic, and suddenly you start looking at papers on pathways, spider web networks, and then suddenly you look up and it's suddenly randomly 5:30 p.m. and wow, did I just spend 3 hours just looking at all of these interaction pathways?
Yeah, no, absolutely. And one interesting thing with the galanin receptors that I'm looking at right now, it has anti-nociceptive activity and has a role in depression and sleep, which are, in a sense, all interrelated. So you could have 3 programs already up and running. One for the anti-nociceptive activity, the other one for the depression, and third one for sleep, and you might end up with a bunch of molecules that do not what you want them to do.
Yeah.
Wonderful, wonderful. So this is your favorite GPCR?
Yes, I would
or
say
the
so.
family
Yes.
of GPCRs.
Yes.
How did you stumble on the galanin family of receptors?
Actually, it was, and I think it holds a special place in my heart because of it. It was the initial first GPCR that Ashish sort of introduced me to, with respect to sort of him trying to tailor projects towards my interest, because I told him that I would be interested in the drug discovery aspect, the structure aspect of it, but also the neurological factor as well. So, and all of those aligned pretty well and pretty nicely with galanin in that respect.
That's wonderful. Can you tell us a little bit at a high level, what are some of the questions you're trying to answer with regards to the galanin receptors?
So I think one thing is just simply, can we find a small molecule for the galanin receptor? And how do we kind of consider that when in the early 2000s, I believe, was when they tried to really explore this receptor a lot more. Various screening attempts were thrown at it, but nothing came up. And that means it's a matter, is it a matter of we didn't dig deep enough? Or is it a matter of the way in which that screening platform was developed? Was that a little flawed, perhaps?
I think, sorry, please go ahead.
No, please go ahead.
I was going to say, I think with the tools that we have today and the combination of assays, add to that the computational power and add to that machine learning capabilities, we are in a better position to ask the tougher questions on these, let's say, more exotic GPCRs than 10 years ago or even 20 years ago. So being able to ask those questions now warrant, I think, going back and looking at all the receptors for which screens were done that came up with nothing 5, 10, 15, 20 years ago and say, well, wait a minute, now we have so much more capabilities, so many more tools. Let's dig deeper into those.
Yes. And to add on to that, I believe it's a matter of needing to integrate those new tools in a way that that enhances the original idea that you had in mind, as opposed to just trying to build the purpose around the tool itself. I think that's kind of that's a danger, I think, when it comes to sort of all of these new exciting tools that we're surrounded by. We have to make sure that we kind of see through the smoke of hype culture, and it's studied carefully and integrated carefully.
I agree. And I think there's a time to ask the tough questions on the exotic receptors and use the available tools in a way that they help answer those questions. But there is also a second bucket when I think about it, is using your, let's say, vanilla type GPCRs. None of them are really vanilla, but the ones that behave in your hands, in order to further develop those tools. And then at some point, there's going to be, let's say, a mix between bucket number 1 and bucket number 2, where you will want to test your brand new tools on the complex GPCR questions. And that's how, in my mind, we are moving forward from a tool development perspective, but also better understanding the role of these more exotic GPCRs.
Yeah, I 100% agree. And it's not even like if you're developing and applying your tools to that vanilla GPCR that you were talking about, it's not as if like it's a waste to develop your tool around those GPCRs. Like, I think there's no better example than the mu opioid receptor. We are always going to need some sort of small molecule modulation of that receptor just because of how prominent it is in both our society as well as our history. So any tool development that is applied on, say, mu or kappa even, or delta, or even nociceptin, really does help push that field itself forward. And now, after that tool is perfected, you can start thinking about other GPCRs that are just much more unknown.
Exactly. And just to clarify, just because I called some of these receptors vanilla doesn't mean that they're not important. I think in order to develop a tool, you need an example system where you can control the output, where you know how your receptor behaves, and use that known receptor behavior to test your tool.
Yeah.
Because with two unknowns, you will never know, is it the receptor or is it my tool?
How would you know, like, how to construct the building if the foundations are not there in the first place?
Exactly. It's a very nice analogy, which gets me to the next segment of our conversation. I'd love to hear a little bit more about the top 3 aha moments that you had as a scientist and that shaped your trajectory. We kind of talked about them, but I feel like at the end of the conversation, listing them out will be helpful for the audience.
So the first one, the first aha moment is definitely that moment where I described earlier, where I sat down the papers September 2022 or 2023 and said something along the lines of, "Ah, crap, I'm going to have to study GPCRs, aren't I?" I think that definitely qualifies as an aha moment. The second one, I think it is just the moment that I was able to show Seth, who is the lead graduate student on that mu and kappa opioid receptor paper that I was talking about. I was able to show him both structures and talk about that small molecule that is bound to both structures and the similarities and the differences between the two. That qualified as an aha moment for me as well, because it was really an accumulation of resolving that initial fear that I had, switching fields, and me realizing that I can do well in this field. It was just a matter of practicing, leaning on the help that is available to me, and the encouragement that the colleagues that surround me, collaborators, as well as my PI, Ashish himself, gave me, that really helped me strive forward and is continuing to help me strive forward of the projects that I'm currently undertaking in his lab. So that's aha moment number 2.
Sure.
Aha moment number 3, I think. I would say it's the moment that I switched from pre-med in undergraduate to the graduate school research / PhD route that I was talking to you about. Because one of my closest friends, Lisa, at the time, she is currently a resident in radiology, and she was telling me, because we were both on the pre-med path, but then I one day decided I can't do pre-med. I don't think I could go through the medical school system. She remarked to me afterwards that she's never seen me pivot so fast of taking the logistical steps necessary to start preparing for graduate school. And this was in junior year of undergrad when I think this happened. So she was absolutely right in that was a fast transition. But I think that really showcased that the path that I was on at the time was definitely not suited for me, but the path that I am on now is definitely suited for me. So I would say that's aha moment number 3.
Wonderful. I'd place that in a chronological manner as aha moment number 1, which is what led you to for us to be recording this podcast episode and having this chat. So I'm grateful for that aha moment.
Yes.
I had a question, and it just escaped me. But let's dive. Actually, speaking of aha moments, I just remembered that question. And being where you are today, I don't know if you have thought about what next after your postdoc, but I'd be curious to hear about how are you framing that question? How are you thinking about that question?
Yeah. So after my postdoctoral research, I think what I would want to do is I definitely would like to remain in the drug discovery field, GPCRs if possible, but obviously I'm not gonna be like tied down to the ideology slash dogmatic nature of it per se. But I think a really fun scenario that kind of describes what I want to do in the future would be like I'm working in a company that is really pushing for a drug candidate, whether it's biotech or a large pharmaceutical company, to which I'm in San Francisco. I can kind of pick or choose depending on what opens up. But I kind of can see myself being the one that kind of looks at all of the data and or the teams that are leading each subdivision, whether we're talking toxicology or preclinical or even manufacturing as an example, and then to be the one that says go or no go. We need X, Y, and Z, or okay, this looks like it's promising. Let me talk to like the necessary people to start the next part of the process. That's something that I can see really appealing to me, and kind of integrating and serving as sort of the bridge between the science that's involved for drug discovery, because that alone is going to be enormously difficult, and then also kind of the steps that need to be taken to really push that into sort of those clinical trials that we're thinking about, and or regulatory compliance and eventual marketing.
It sounds like you have your work cut out for you. After this, which is great because I think it's important for you to know what direction you want to go into, and then look at what's out there and match those, let's say, aspirations to a specific job title, and then you can enter that, let's say, road in that field based on, okay, this is what I want to do. Here are the things that I need to learn in order for me to do that.
Yes, and when people say I have my work cut out for me, I kind of take that more along the lines of kind of going back to the theme that we covered earlier. I'm not going to get bored. That is for sure.
No, I agree. And it would be boring to do something that you've already, mastered and know how to do. And at some point you just don't enjoy it anymore. So that need to learn something new, yes, I can actually very much relate to.
Yes.
What is your advice to junior scientists who are aspiring to contribute to the GPCR field?
Yeah. So one advice that immediately stands out to my mind is don't let fear stop you from pursuing the crazy ideas that you are thinking about, or even just entering the GPCR field itself. So not only, as an example, not only did I enter the GPCR field based on my realization of drug discovery and membrane proteins, but I kind of gathered a lot more like data / information, and that was really helpful in kind of helping me transition from what used to be a very non-protein biochemistry field. I was mostly working with cells and neurons per se, to the GPCR field that I am currently in right now. It's perfectly fine to feel fear, but it's not going to be so helpful if you let that stop you from that pursuit.
I agree. I think the worst thing that you can do is nothing.
Yes.
Any action is better than not doing anything. You might not do the right thing from the beginning. You might stumble, but you will learn from that, and that is going to help guide you towards the right direction.
Exactly. And that leads me to the second advice that I would want to give, which is like try to understand or ingratiate yourself in an environment where that sort of fear is perfectly fine to have, and you have the space to learn from that.
Yeah.
And that was like a large part of why I ended up going with Ashish's lab in the end, out of the offers that I had, because I knew, mostly because I really emphasized this to Ashish, that I am very afraid of entering this GPCR field. But hey, I entered the environment that allowed me to feel that fear, but I was able to quote unquote master it. And the success that I've had since then is really a sign of being in that environment that I was talking about, where I don't have to worry about stumbling and then falling down completely flat on my face, per se. There were people around me that picked me up and said, yeah, okay, like, sure, there was a mistake or two, but we can do X, Y, and Z next time. And I did that X, Y, and Z, and hey, it turned out okay.
I agree. Joe, thank you so much for your time. I really enjoyed our conversation.
Thank you, Yamina. It's been an absolute pleasure.
Thank you. Don't go anywhere.